In the preceding lecture with the TAs, you were introduced to the core processes of transcription, the actual mechanisms of transcription, elongation, assembly of the pre-initiation complex before that, termination, cap modification, polyadenylation, splicing, etc. The focus of today's class is going to be on how transcription is regulated. What are the key mechanistic steps that occur upstream of assembly of the pre-initiation complex and downstream of transcriptional events that dictate the outcome of any gene regulatory circuit or pathway in cells. As with the core elements of transcription themselves, we're going to start off by talking about transcriptional regulation in prokaryotes, and then build off of that to talk about eukaryotic transcriptional regulation. And what we'll begin with is a classic example of gene regulation in bacteria related to the utilization of lactose. All right, so lactose is a disaccharide. It's a sugar, all right, milk sugar, as well as from elsewhere. That can be used as a nutrient source for bacteria in their metabolic pathways. We've not covered metabolism yet. That's quarter 4. But you can use the lactose as a carbon substrate to generate ATP and use it for the building blocks of life. For that to be able to occur, if there's lactose in the environment of the prokaryote, you have to be able to get the lactose into the cell to be able to metabolize it. And so what occurs in bacteria is that lactose, chemical structure is shown here, large-ish, hydrophilic, so it's not going to go through a membrane just by passive diffusion. It needs a dedicated transporter to move it across the membrane, and this is achieved by a transporter called galactoside permease. This is a transporter that will move hydrophilic solutes across the hydrophobic membrane. Once inside the cell, lactose is broken down into 2 monosaccharides by an enzyme called beta-galactosidase that cuts right around here, and then will generate 1 molecule of a monosaccharide, glucose, and another molecule of the monosaccharide, galactose. And these can be metabolized through pathways that we'll talk about in a couple of weeks. What our focus on today to begin is that the galactoside permease and the beta-galactosidase are both regulated transcriptionally depending upon the metabolic state of the cell, as well as the availability of lactose in the environment. And its regulation occurs intimately by transcriptional processes that we're going to expand upon in a moment. What is evolutionarily advantageous for the bacterium is to only express the enzymes such as beta-galactosidase only when they're useful, meaning that when lactose is available for them to have substrate to turn it over and fuel metabolism. If there isn't any lactose or metabolic conditions are more favorable to use another carbon substrate, it's energetically wasteful to make beta-galactosidase or the permease simply for the sake of making it. There's a cost that's associated with those. And so this is the evolutionary driving force for these transcriptional regulatory pathways that we're going to talk about. In prokaryotes, a common conceptual model for transcriptional regulation that has a mechanistic basis to it is called the operon. All right? And what are operons? Operons are compact sets of genes, right, that are all contiguous with one another, next to one another on the prokaryotic genome, and in fact are transcribed as a single mRNA. So single pass RNA polymerase transcription encodes all of the genes for one of these operons on an mRNA. And that mRNA is so-called polycistronic, and what that means is it is translated at multiple points along that mRNA to give rise to the protein products of the genes that just got transcriptionally induced. I'm going to say not too much more on that because we haven't touched on translation explicitly, but these polycistrons effectively have places for ribosomes to bind at multiple places along the length of the mRNA body to begin translation, make the protein product, fall off, and then dock somewhere else on that long mRNA to encode for protein. Polycistrons and operons are unique to prokaryotes and also some viral genomes will have a polycistronic mRNA. We don't have them, although if they're presented to us, they will be used by our host ribosomes. On the bottom of this slide, what is shown is the operon related to lactose metabolism, imaginatively called the lac operon, and it's shown schematically here. You talked about the promoters, gene promoters last time. Here is the promoter for the lac operon. I'll speak more about this operator element in a moment or two. But downstream of the promoter and the operator are the genes, the lac genes that comprise the operon. You have lac-Z, which encodes for beta-galactosidase. Then lac-Y, which is the permease, all right, so the transporter that moves lactose into cells. And then lac-A is another enzyme. It's a transacetylase that acetylates beta-galactosides, even though it's not absolutely required for the metabolism of lactose. So it's a little bit mysterious exactly why it's in the operon. So we mentioned it here for completeness, but our major interest is on how this operon gets turned on in bacteria as a function of the environment of the cell. Operons and the genes that are encoded within them can fall into different categories. We call structural genes are genes that in one of these operons that are directly important for the function that the operon is related to. For instance, the permease, the galactosidase. Right? These are enzymes or transporters that are directly involved in the function of lactose metabolism. These stand in contrast to regulatory genes that encode functions related to the induction, the turning on, or the repression, the turning off of the operon itself. Hopefully that's self-explanatory by the name. Structural genes and regulatory genes can be in an operon. They also can be outside of the operon, and especially the regulatory genes, and impinge upon the operon itself. The weaving of structural and regulatory genes into common operons, if people have heard of synthetic biology, we may have, I'm not sure if we've mentioned it yet up to this point in the semester, but when one tries to apply engineering principles to design biological regulatory circuits, a lot of them are geared with operons and embedding regulators in the operon that may control the transcriptional regulation. So an understanding of what's the business end of the operon, meaning the structural genes, and what are the regulatory genes related to the operon is an important distinction. Let's speak more about the operator. And so an operator sounds similar to operon, but it is distinct because now we're talking about a gene sequence, a gene element that's going to be close to the promoter, but before the first gene that's encoded, structural gene encoded. You can think of this almost like a landmark or potential roadblock to transcriptional induction of the operon itself, depending upon what is bound or has the potential to be bound to the operator. It is a DNA element that has the capacity to alter the transcriptional activity of that operon. The regulatory gene that's related to the lac operon it happens to be adjacent to the operon itself, but I want to emphasize that it's not in the operon proper. It's just, they show it geographically, it's close by, but it's not actually in the operon. Question? So the regulatory. Having the microphone on would be good. All right, thanks. I thought I was projecting well, but not as well as this. So the operon for lac, encoding these structural genes. I want to zoom in now on this regulatory gene called LAC-I, which is over on the left-hand side of this schematic, but not in the operon. But this is where the regulatory gene is encoded. So the lac operon itself is regulated by a transcriptional repressor, which is this LAC-I. What a transcriptional repressor does is it serves as the protein recognition element for the operator, and that as a result of binding to the operator in the operon, it inhibits or blocks transcription. So this is a negative control of transcription. When it's bound, you can think of it as a roadblock that prevents RNA polymerase from being able to go through the operon and transcribe it. And I think the one thing that I also wanted to add here is that as shown by this schematic, one of the major means by which these transcriptional repressors operate in bacteria is by physically blocking the place where RNA polymerase would ordinarily bind. So it's a physical exclusion, largely, of RNA pol2 to be able to elongate. So no binding, no transcription. Now the LAC-I gene encodes for a repressor called the LAC repressor. And the interesting characteristic of the LAC repressor is that its repressive ability is determined as a function of the available lactose inside the cells. When there is no lactose inside the cells, the LAC repressor serves as a repressor. It recognizes the operator and inhibits the operon. However, when lactose is around, cellular lactose is around, it binds to the LAC repressor. That LAC repressor undergoes a conformational change, an allosteric change, that then prevents the LAC repressor from serving a repressive function, no longer can recognize and bind to the operator. And then you'll see what the consequences are of that here. So now let's say we're in a scenario where there's lactose around. So the cell ordinarily would be in this inhibited state, no induction of the LAC operon because the LAC repressor is fully active, bound to the operator. When lactose goes into the cell, it will bind to the repressor, undergo a conformational change. Now it's no longer binding to the operator. RNA polymerase will then bind, transcribe the operon, and now you have the mRNA for LAC-Z, LAC-Y, LAC-A. This mechanism of transcriptional regulation is pretty simple, and it occurs repeatedly in broad strokes terms, both in prokaryotes and eukaryotes, and it is referred to as substrate induction. And so if you have a metabolic pathway where you need to encode a series of enzymes, or even one critical enzyme, to be able to act upon a particular substrate, if that substrate is able to transcriptionally upregulate the enzyme that will metabolize it or convert it to product, this falls categorically into the definition of substrate induction. If we get down more into the details of exactly what is happening here, in the LAC operon, its regulation is occurring by derepression. You're inhibiting a repressor, so inhibition of a repressor will serve to turn on the operon. You heard earlier in the semester disinhibition? Yes, good, all right, thank you. Yeah, so disinhibition is a negative of a negative is therefore a positive. So this is a form of disinhibition or derepression. So we have substrate induction, an example of the lac operon. What does the lac operon do? Once these enzymes are expressed in the cells, now you have an enzyme to be able to break down lactose. So this breaking down of lactose as a carbon source is an example of catabolism, or a catabolic pathway. So taking complex biomolecules, breaking them down into simpler subunits is called catabolism. Cells also will build up more complicated biomolecules from simpler precursors. This is called anabolism, or anabolic pathways. And so the next example of transcriptional regulation in prokaryotes that we're going to talk about involves an anabolic pathway related to tryptophan biosynthesis. Tryptophan, to remind, is one of those hydrophobic amino acids that you learned at the very beginning of the class. It is somewhat complicated of a side chain to make biosynthetically, and therefore bacteria have a series of enzymes that perform the chemical modifications on amino acid precursors to enable the cells to synthesize tryptophan. It's also energetically costly. You only want to be expressing those enzymes when there's a need for tryptophan. And when there's no longer a need for tryptophan, the cell would like to stop expressing those enzymes because there are several of them to be able to engage the anabolic pathway. This gets us to what's called the trip operon, trip for tryptophan. The operon is shown here schematically on the right-hand side. There's a promoter. Here's the operator. For the purposes of today, you can ignore this leader sequence. It's just a little bit of a separation of bases here before you have the 5 genes for tryptophan biosynthesis, shown here, TRPDECBA. So what is the repressor for the trip operon? It's the trip repressor, as shown here. This schematic is also geographically accurate. The regulatory gene here for the trip operon is elsewhere in the bacterial genome. So it's somewhere else, not in the operon, but what it encodes is the trip repressor. And the trip repressor has an inverse regulatory relationship compared to the lac repressor. Its ligand, or the small molecule that can bind it, is tryptophan itself. And to serve as a repressor, the trip repressor needs to be bound to tryptophan. Thus, in the absence of any tryptophan, the trip repressor is inactive. Okay, so the operator is unbound. The promoter is available for RNA polymerase to go in and transcribe. However, once that biosynthetic pathway has made enough tryptophan, or there's enough in the environment for it to no longer be necessary to synthesize tryptophan, the excess tryptophan is going to bind the trip repressor, and now you have an active repressor that can recognize the operator on the trip operon and block RNA polymerase from transcribing. You think, they all involve small biomolecules, they all involve repressors, but think about the regulation of this. This now, the downstream output of the pathway, the consequence of the pathway, is feeding back onto the regulator that induces it in the first place. All right, and so this is a different regulatory mechanism that we call end product repression. The net result of the induction of the operon, or the induction of the different genes, creates a product that feeds back onto the transcriptional regulatory machinery and shuts it down. It's no longer needed. End product repression. Yes? And so the question is, does this not happen in eukaryotes because of the segregation of organelles? You have genes in the nucleus, and then you have other, like a lot of metabolism happening in the cytoplasm. It can happen here. Part of my hesitation is it's that similar theme where we don't have operons, we don't have big contiguous subunits of genes that get transcribed at the same time, but metabolites, small molecules, information exchanges between the cytoplasm and the nucleus a lot, very regularly. We'll have more to say about that in quarter 4. The theme of negative feedback regulation, where a downstream product shuts off the enzymes or other pathways that gave rise to that type of negative feedback regulation with transcription occurs just as much in eukaryotes as in prokaryotes. It just might not necessarily involve, or it's not going to involve operons. There could be more genes involved, and it's a bit more complicated. Yeah, excellent question. So, okay, well, we talked about the regulation of the operon. What about these regulatory genes? How are they regulated? Note in this cartoon and in the other one before it, TRPR has its own promoter as well. And that promoter can be subject to other regulatory inputs. It's abstracted here, and I actually don't know the details on this particular repressor. Sometimes these ones may be transcribed at a basal level because most of the sensing and the regulation of the pathway occurs at the molecular recognition, in this case of the TRP repressor, and the availability of tryptophan. So that's where a lot of the regulation occurs, more so than the abundance of the TRP repressor, but you could have more elaborate ones as well. All right, to review this part, we talked about two different operons. Lac operon, where you have the substrate for the this enzymatic pathway that will be catalyzed by the genes encoded in the operon, are binding to the repressor and disinhibiting it, de-repressing the operon, allowing transcription to occur. And then with the TRP operon, it's the downstream product rather than the upstream substrate that is feeding back onto the repressor to shut it down when it's no longer needed anymore. I have this slide to get home the point that these two both involve repressors. All right, so they're transcriptional repression events. Are regulated differently, and they have different consequences, but they're repressive. They don't always have to be this way. There are also, if you will, positive types of transcriptional regulation, even involving one of the operons that we've just been talking about. In the lac operon, there are other regulatory inputs to how it is controlled. And one of the key additional factors that determines whether the lac operon can be induced is the availability of a preferred carbon source, glucose. More to come. Quarter 4 on why glucose might be a preferred carbon source for metabolism. But first characteristic is that the enzymes needed for glucose metabolism are always on. They're on and they're always available. They're ubiquitously expressed, constitutively expressed. So always being transcribed and translated. Because of that, if there's glucose around, and you can make downstream metabolites and energy from them, why not use those enzymes that are already expressed? And so if there's enough glucose in the environment, you would like not to have to turn on other ones around if you have your preferred carbon source. Something like the lac operon, from an efficiency perspective, should only be transcriptionally induced when there is a metabolic need by the cell. And if you think about this simply from the perspective of lactose and glucose, you want the operon to be turned on only when glucose is low and when lactose is high. So we need to get to how the glucose low or the glucose state could determine whether or not the lac operon is regulated. And the way that this occurs is on another DNA element close to the promoter for the lac operon called the CRP site. And CRP stands for catabolite regulation protein. And this is a catabolic pathway. That's the reason why it's called the C part. And CRP has an important characteristic. It recognizes another small biomolecule, one that you've heard before, second messenger, cyclic AMP. In prokaryotes, cyclic AMP and glucose have an inverse relationship with one another. If glucose is very abundant, cyclic AMP signaling is lower, and then vice versa. As glucose goes down, cyclic AMP goes up. So let's go in the scenario of glucose being low, cyclic AMP being high. When cyclic AMP is high, it's high enough that it will bind to CRP and form a bimolecular complex. Similar to small molecule protein interaction, just like lactose and the lac repressor, tryptophan and the tryp repressor. However, in contrast, when CRP binds cyclic AMP, that complex, it gets bound to the CRP site in the vicinity of the promoter. And this complex binding nearby to the promoter for the lac operon is required for RNA pol2 to bind and then transcribe. So if you don't have it, RNA pol2 can't come in. It doesn't matter the state of the operator lac repressor. So you need to have that to open up the possibility of lac operon transcription to occur. And so for time, I'll just leave this as a thought question. If you guys want to talk it out over Piazza or something like that, you can run through the conceptual scenarios of what happens when you have glucose on, lactose low, lactose high, both high. And you can see how these two forms of regulation can converge on the lac operon. All right, we're almost through the prokaryotic part. We need to talk about a couple more terms that come more to the fore when we begin discussing eukaryotic transcriptional regulation. And that is this theme of DNA sequence in the vicinity of where transcription occurs, in the local vicinity of where the gene that is getting transcribed. And so this local vicinity, the immediate proximal nature of these DNA elements are called cis-acting elements. So the operator, the CRP site, these are cis DNA elements. All right, so they're DNA, which we're referring to, and they're cis because they're right on the strand where the transcription occurs. In converse, when we talk about regulatory factors or regulatory genes, things like the lac repressor, the tryp repressor, CRP, they're commonly encoded elsewhere. The lac is a little weird because it happens to be close by, all right? But the key thing is that the way in which they impinge upon the operon is by a protein product coming in and then binding onto the DNA nearby where the gene is transcribed. Because you have that more distant relationship between where the DNA element that encodes the factor and then where the binding, this is called a transacting mechanism. If you want to talk about the DNA that encodes those regulatory genes, it's a transacting element. But the protein products are called transacting factors. So the theme here is that what causes a factor to be in trans is that you need to have that protein be expressed. It could be expressed anywhere. Then it diffuses, recognizes a site, a cis-acting element, an operator on the DNA sequence right where transcription will occur. So transacting factors acting on cis-acting elements. We clear on these terms? Yeah, so you're asking clarification on the one seemingly confusing part in that the LAC-I gene happens to be close by to the operator. It's still a transacting factor because it is transcribed separately, separate promoter. We talked about what are the promoters that control the regulatory genes. And then the way in which it impinges on the operator is by protein diffusion, and it goes on. Another way you could think about it is I could take that lac repressor and put it elsewhere. I could put it on another plasmid in the bacterium, and it would still serve as a transacting factor. Whereas if I took the operator sequence and I took it out of the operon and I put it somewhere else on another piece of DNA, it's not going to execute its function. Maybe that's a way to think about it conceptually, about what determines the cis versus trans nature of factors and elements. Good. Now that we have the basic concepts of transcriptional regulation in prokaryotes, we can now pivot to the similarities and differences in eukaryotes. We also have transacting factors, transacting proteins, which we call transcription factors. And these bind to and recognize cis-acting elements, and those are DNA elements. So that's all the same. The complexities is that the distance between your definition of cis and these binding events are not as compact and as local as I introduced you for prokaryotes. I have a schematic of this in a moment. And then the other thing is that this very clean one-to-one relationship between the trip repressor and the trip operon, or the lac repressor and the lac operon, that breaks down heroically in eukaryotes, in that we have many different transcription factors binding to many different DNA elements all across the genome, each of which is impinging on the transcriptional regulatory state of each one of those genes. And so this is a lot of the complexity of eukaryotic transcriptional regulation. I'll walk you through an animation of this. Let's say we have a stimulus A, and we have two genes here. We're not in operons anymore. These are just single genes in a eukaryotic genome. But we have a stimulus A that causes the expression of one of these transacting elements, giving rise to a transacting factor, a transcription factor, that we'll just call transcription factor A for the purposes of this cartoon. So transcription factor A recognizes a cis DNA element on these two gene loci shown on the bottom here. They don't have to be close to one another, they're separate, okay? But there are elements upstream of their sequences for binding for the transcription factor A. Let's say that concurrently cells experience another stimulus, stimulus B, that induces another trans element, another transcription factor, but now only the gene on the left-hand side has the DNA element that's recognized by transcription factor B. What occurs now in cells, to a first approximation, because the more of these transcription factors, activating transcription factors that are being recruited to a DNA locus, the more efficient they're going to be at assembling a pre-initiation complex, the more efficient that they are going to be at recruiting Pol2 to begin transcription. The net result is that because you have more Pol2 occupancy on the locus where you have multiple transcription factors bound and recruiting, the first prediction, the inference is that you'll have many more mRNAs being transcribed off of the locus on the left compared to the locus on the right, simply by the multiplicity of transacting factors binding onto the DNA elements and aiding in the formation of pre-initiation complex and the recruitment of Pol2. All right, so more TF binding, to a first approximation, is more polymerase recruitment and more transcription. My pause is because there's always exceptions to the rule, but generally people think more binding is going to be more activity when you're talking about a classic transcription factor. And more to say on these cis control elements. I'll start by orienting you around the things you've already seen before: Tata box, initiator sequence, core promoter, transcription start site, and then you're into the gene. In eukaryotic cells, we can have, so when we talk about upstream, we mean if this is position 1 here of the transcribed gene, we're going 5' on the coding strand from where the transcription start site is. So that's what we're talking about, we're upstream. Within 100 or a few hundred base pairs of that transcription start site, you can have DNA elements that will recruit transcription factors. These are called proximal control elements. They tend to have a very potent effect on the gene regulation in the vicinity of where that transcription start site is. But the added complexity is that we in eukaryotes have a large number of hard to predict and hard to identify, except by experiment, distal control elements that can be hundreds or thousands or tens of thousands of base pairs away from the transcriptional start site that they regulate. In addition, these distal control elements can have an enhancer function. They can promote transcription factors that aid in the formation of the pre-initiation complex and Pol2 recruitment, or they can also serve as repressors. They can displace other transcription factors that might serve as enhancers, or they can interfere with the ability of the pre-initiation complex to form. They can recruit other factors that just shut down that DNA from being available for transcription. So there's a lot more back and forth on positive and negative regulation. But the key complexity is that you're no longer dealing so cleanly in the linear sequence space. In prokaryotes, because all of the regulation is very close to where the transcription start site is and where the promoter is, you can kind of just look at it, you draw it out like a big spaghetti strand, and you read off the things. In eukaryotes, where a lot of this assembly here, where you have enhancer elements bound by transcription factors, they help in the formation of the pre-initiation complex, where all this looping and the bending of the chromosomes, the thinking is that it's not like it's bent out like a straight stick. The binding happens and then it folds over like this. In reality, this locus was already contorted like the spaghetti strand that you see here, and the elements were already in close 3D proximity of the transcriptional start site that they end up regulating. And so there's a non-linear 3D topological arrangement of these enhancers and the transcription start sites that they regulate that is very difficult to predict, pretty much impossible to predict by bioinformatics, but we can measure these types of things by experiment. I'll give you a real-world example of this. This is from my own lab, where we were interested in these two transcription factors called FOXO and RUNX. They recognize certain DNA cis elements. They're shown here. The R's and the Y's, those imply different combinations of A, C's, G's, or T's that are permissive in that site. But for instance, it's got to be a certain number of bases that fall into this R categorization, T, A, Y is another set of them, and then A. These are indicated by the upside-down green triangles. These are a set of genes that we were interested in. Here's the transcription start sites. Ignore the purple parts. That's how we measure them experimentally. But look at all of the upside-down triangles that have DNA sequence that conforms to a cis element for recognition by either FOXO or RUNX in green and yellow. Some of these could actually serve to recruit Pol2 and assemble the pre-initiation complex. Some of them are bound and some of them are active, but a lot of them are not. They're not accessible to those transcription factors. And so what we need to do in the lab is go in and experimentally try to figure out where are these actually getting bound by the transcription factors, which ones do we think have the strongest chance of regulating transcription of the gene. And so there's a lot more complexity when you look at eukaryotic promoters compared to prokaryotic ones. And unfortunately, I can't say anything more satisfying than that. In essence, what the field is doing is coming up with more and more elaborate ways of measuring them empirically in the hopes one day that we can learn more about making computational predictions or inferences about which elements are really critical to regulate which genes, but it's very much in an empirical phase right now. But we have enough raw materials now to turn back to the original disease motivation and talk about mature onset diabetes of the young. Sham and Emily talked about where this falls into the broader spectrum of diabetes. Here expands upon this a little bit about the different subcategories or classes of MODY. And what I want to emphasize are these two classes that together, about 3/4 of all cases, related to a gene and protein product called HNF, either 1 or 4. So how does HNF relate to MODY? First, what does HNF stand for? HNF is a transcription factor. It stands for hepatic nuclear factor. It was originally identified in the liver, but these HNFs are expressed broadly in us. And HNF is in the category of the transcription factors like I just talked about. However, HNFs have another partner in them called DCOH, dimerization cofactor of homeodomains. It acts, if you will, as a bridging molecule to bring together HNFs and to recruit the other RNA, Pol2, and other factors of the pre-initiation complex. It's the trigger for assembly and transcriptional activation, whereas the HNFs provide the DNA recognition. To expand upon that, HNFs, transcription factor, but meaning they recognize a DNA sequence, they recognize an element and bind to it, but that ability to recognize and bind to DNA only occurs when it's a dimer. And you'll see why structurally in a moment. The dimer gets held together by DCOH. And so it's a mixed assembly of HNF and DCOH. And only when you have DCOH with HNF can you get the recruitment of all of the other components of transcription, which is what I just said. So let's show a cartoon of HNF and DCOH on a locus that's particularly important for blood sugar control and diabetes, and that's the expression of the insulin gene. Here's the gene, here's the promoter region, and so in this cartoon, we'll assume that there's a proximal element that's important for HNF. What ordinarily happens when insulin regulation occurs normally is that when HNFs get expressed, they will form a heterotetramer, 2 HNFs, 2 DCOHs, as shown in cartoon form here, and then this heterocomplex will recognize the HNF DNA elements in the promoter of the insulin, and that recruits pre-initiation complex and the RNA Pol2, and then you turn on transcription, making mRNA. In Modi cases, you have an inherited mutation. Here's the ribbon diagram, 2 DCOHs, 2 HNFs, and the DNA is going to wrap through here. It's not shown in the cartoon, but you need to have these two going in around, and that recognizes the DNA sequence. Where these mutations occur are right at this binding interface here. So the 1 and the 4 here prevents the HNFs from interacting with the DCOH. You no longer can form the dimer is being brought together by the DCOH. So you don't have the DNA recognition anymore, no longer able to transcribe the insulin gene as effectively as normal. The green hexagon and the yellow circle here, this would be your basal transcription factor, TF2D, right? And that was the abridged version of the pre-initiation complex, and then recruitment of Pol2 and transcription. TF2D, Pol2. Okay. So pretty straightforward. You break the transcription factor. You break the ability to serve as a recruitment of all of these other components of the pre-initiation complex and Pol2. Separated from its ability to recognize DNA, you don't get proper regulation of insulin. In contrast, even though this is there's a problem with blood sugar control and a problem with insulin, these patients are not insulin resistant. They just don't have enough insulin in there. So they can be treated with insulin. There are also other orally available compounds to try to lower blood sugar. I list them here for time. I'm not going to expand upon them. And then the last thing that I'll say is that although there remains a lot of excitement for transplanting islets to be able to bring in pancreatic beta cells that would have normal transcriptional regulation and be able to express insulin, this still remains very highly controversial, or not controversial, highly experimental treatment, expensive, and it's still undergoing research. In the final couple minutes, what I want to talk about is the other beginning of the wrapper here for transcriptional regulation related to steroids. Also involve transcriptional regulation, but very different from the disease like I talked about before. A couple minutes on steroids themselves. The chemical structure of several representative steroids are on the right-hand side here. What they have in common is what's called the steroid nucleus. This 1, 2, 3, 6-membered ring adjacent to a 5-membered ring. You need to be able to recognize this. It comes up again and again in molecular biology. You can look at it and you can broadly categorize what is has the potential to serve as a steroid hormone in cells. Cholesterol has one. So do things like testosterone, estradiol, et cetera, shown here. The key thing is, if you look at the chemical structure, it's all hydrocarbons, okay? Hydrophobic. So the hydrophobicity has one advantage and one consideration. One advantage is because of its hydrophobicity, it doesn't need a transmembrane receptor to be able to go through a plasma membrane. It can just diffuse through because it's hydrophobic. However, to be able to circulate in the bloodstream, it needs to be bound to something that has a hydrophobic character to it because it's an aqueous environment. And so that's achieved by the abundant blood protein albumin, which serves a number of different functions in our circulation, but one of them is to transport hydrophobic molecules. Even though steroids don't have transporters, or sometimes you might call it or some type of receptor on the plasma membrane, they still have receptors. They're just inside the cell, not on the plasma membrane. And so here's the gist of how steroid hormone pathway works. This is shown specifically for glucocorticoids, but the mechanism is straightforward and is thematically similar for all of the steroids. Steroid here, outside the cell, diffuses through the plasma membrane. Intracellular receptor for that steroid. Here, a couple of different things can happen. One, some of these steroid receptors are bound to an inhibitor that keep it in an inactive state. Others are simply in a conformational state that is inactive. It doesn't involve another protein recognition. These steroid receptors, when they bind to their steroid ligand after it has gone through the plasma membrane, if it's bound to an inhibitor protein, that inhibitor will dissociate. If it's in a conformational change that's ordinarily inactive, they'll undergo a conformational change and become active. And so by virtue of some form of disinhibition, either unbinding of the inhibitor protein or conformational chains, now you have an active steroid receptor that will translocate from the cytoplasm into the nucleus. These also can dimerize to serve as a transcriptionally active unit. There are steroid response elements, DNA elements in our genome that serve as the docking sites for these steroid receptors. They dock onto those DNA elements and then will promote transcription in the vicinity of where they bound. So we were going to talk about like muscle building, steroids, and things. This is one example of one of these artificial steroids. It's based off of it's a ligand that serves as a very potent factor for the androgen receptor. The normal ligand for the androgen receptor is androgen or testosterone. So it's a superpowered testosterone. Chemical structure is shown here. See the steroid core. So the two things are why you have this as a synthetic steroid and is a very potent activator is twofold. One, it has a higher affinity for the steroid receptors than testosterone itself. And second, by some of the synthetic modifications, it doesn't undergo some of the feedback regulation that ordinarily accompanies testosterone signaling in cells. So it's kind of on all the time. So what happens now? AR, in the presence of these steroid hormones, is active all the time inside the nucleus, turning on genes. And one of the major targets for androgen receptor signaling is not insulin itself, but another ligand in the family called insulin-like growth factor, or IGF-1. And IGF-1 is a diffusible hormone, a peptide hormone. It has a transmembrane receptor. It activates cell signaling inside the cell. And what it does is promotes protein biosynthesis and cell growth. So translation gets revved up. Cells get bigger. Muscle cells get bigger. All right, so more sarcomeres, cross bridges, and all those other types of things. And concurrently, it downregulates catabolic pathways, things that would break down proteins inside the cell. Those are similarly inhibited. This is the reason why, just like certain hormones, IGF has been used for growth failure, or people will use it for a performance-enhancing agent. And the last bit of info on it, if there's any pet lovers here, IGF and the abundance of IGF is a critical determinant of your dog size. All right, so Great Danes, lots of IGF just because of the way that they've been bred. Little Chihuahuas, little bit of IGF, and that same holds for us. And so if you're interested in transcriptional regulation, David, Marty, and David, all in this building in BMG, that's what their labs work on. And I'll see you on Thursday.